All right, we're continuing on here with our study of the Ciliophora, phylum Ciliophora, the ciliates. And if you'll recall, we're looking at paramecium as an example. And this is a great image of paramecium. This is a scanning electron micrograph of a paramecium. You can see just how hairy they are with all the cilia all over the outside of the cell. And you can very clearly see the oral groove here where food is funneled to the gullet at the bottom of the oral groove. But the purpose of this slide is partially to learn about the structure of cilia and flagella. Cilia and flagella have the same structure. If you look at a cross-section, and that's what we're looking at in this transmission electron micrograph, we're looking at cilia that have been cut, and we're looking at them on end. And you can see the arrangement of microtubules, but you really can't see it that easily in this micrograph. So here's a closer view of a transmission electron micrograph through a sperm flagellum. So again, this shows that flagella and cilia have the same microtubule structure, and we call that a 9+2 arrangement of microtubules. If you'll recall, we were also looking at the microtubule arrangement of centrioles in animal cells, and they have a 9 times 3 arrangement, where there are 9 triplets of tubules going around the outside, and there's nothing in the middle. But in this 9+2 arrangement of microtubules in both cilia and flagella, there are 9 pairs of microtubules around the circumference of the cilia or flagellum, and in the middle there's another pair. That's the +2. So 9 pairs around the outside plus 2 in the middle. Not 2 pairs, just 2 microtubules. So they just refer to this as a 9+2 arrangement. So protozoans reproduce asexually by mitotic binary fission. And you have to specify mitotic because, if you'll recall, prokaryotes also reproduce by binary fission. Binary fission just means splitting in 2. But in prokaryotes, they don't have a nucleus. Therefore, it's not mitotic binary fission. Mitosis is not involved. Mitosis being the division of the nucleus. Well, if there is no nucleus, then there's no need to divide the nucleus, and prokaryotes don't have a nucleus. So they do copy their DNA before they divide, but they don't have to divide a nucleus because they don't have one. But protozoans do. So protozoans reproduce by binary fission, and that's what we're seeing going on here. And here's closer view. So here's the nucleus, and it has divided, and now the cell is dividing, and that's known as cytokinesis. So complete cell division is a 2-step process: mitosis and then cytokinesis. Conjugation is what's going on here, and conjugation is different in eukaryotes than it is in prokaryotes. If you'll recall, prokaryotes can also conjugate by forming a pili bridge and passing a copy of their plasmid across a pili bridge. But in eukaryotes, meiotic copies of the micronucleus is passed between individuals that have fused their plasma membrane together and formed a bridge, a cytoplasmic bridge, connecting their cytoplasms. And they pass copies of their micronucleus and exchange copies of their micronucleus that were formed through the process of meiosis, not mitosis, but meiosis, which is the same process that forms sperm and egg sex cells. So it reduces the number of chromosomes in half and also produces variation in those sex cells, those sperm and egg. But in this case, no sperm and egg are being produced. This is not sexual reproduction. This is just the exchange of genetic information between 2 cells. The reason for this is because as paramecia age, as protozoans age, damage takes place because of multiple binary fissions. You know, they just split in 2 and split in 2 and split in 2 and split in 2, and with each division there's a little bit of damage done to their DNA. And there's also damage done to the DNA by ultraviolet radiation and other environmental factors. So basically they age and they become sickly, and that's when they need to conjugate. They conjugate in order to kind of revive themselves and make themselves young again. You can think of it that way anyway. So the result is restoration of the mutated genes, restoration of those genes that were damaged by all this binary fission going on, and an increase in genetic variation and diversity, because again, meiosis is how the micronucleus divides prior to the exchange during conjugation, and meiosis produces genetic variation and diversity, which you know is very important. It's the raw material of natural selection and evolution. So here we're looking at the details of conjugation. And so it begins when two old, quote-unquote, old paramecia that are sickly and damaged from binary fissions, form a cytoplasmic bridge. They fuse their plasma membranes and form a cytoplasmic bridge. Their micronuclei undergo meiosis. So here's the micronucleus in both the individuals here, and they undergo meiosis, and meiosis produces 4 micronuclei. The result of meiosis of one micronucleus is 4 micronuclei in each individual. 3 of them disintegrate, however, in this process, leaving only one behind, but then the one that's left behind replicates by mitosis, so that each individual has an exact duplicate of the same micronucleus, and then they exchange those. They exchange one between them, so that each then has 2 different micronuclei. Those 2 different micronuclei then fuse together and form one diploid micronucleus. And by the way, I didn't mention what haploid means here. Haploid means half the number of chromosomes, and diploid means the full number of chromosomes. So these micronuclei, after they undergo meiosis, only have half the number of chromosomes. And the micronuclei, before they fuse together, they still have half the number of chromosomes. But once they fuse, that returns the number of chromosomes to the full number that are supposed to be in this species. In the meantime, as you can see here in the diagram, the macronucleus disintegrates. So the macronucleus is the one that had damaged genes, and is the whole reason that these 2 individuals needed to conjugate in order to repair those genes. The result of this process is that it revitalizes the cells. Basically, they become young again. They're genetically identical to each other because they've exchanged genes with each other, and they've mixed those genes together. They've shuffled those genes together into a new combination, but they both have that same combination of genes. But they're genetically different than all other paramecia. They're genetically unique, so that introduces variation into the population. So conjugation, as I mentioned, is not a form of reproduction. It doesn't produce new individuals. We started with 2 paramecia and we end with 2 paramecia, but what it does do is introduce variation and revitalize them. So the purpose for the individual, for these individual paramecia, is revitalization, to repair the DNA damage that was done by all those binary fissions. For the population, though, of paramecia, the important thing is the introduction of genetic variation and diversity. All the gene shuffling that goes on during meiosis, and then the combination of the genes between the 2 individuals produces genetic variation and diversity, which again, as you know, is very important. It's the raw material of natural selection and evolution. If there's no variation, there's nothing for natural selection to work with, and therefore species do not evolve and adapt to their changing environment. So the bottom line is they become young again and genetically identical to each other, but that's only 2 cells in a population of who knows how many paramecia, but they become highly variable compared to those other paramecia. So that's it for our look at the phylum Cilia. Now we're going to move on to the other phyla of protozoa, starting with the phylum Zoomastigina. So here's the phylum name, Zoomastigina, but they're commonly referred to as zooflagellates because they have flagella. And that's what you need to remember about zoomastiginids, is that their mode of locomotion is flagella, just like the mode of locomotion for the phylum Ciliophora is cilia. And cilia and flagella are closely related to each other in terms of their structure. They both have that 9+2 microtubule arrangement in their ultrastructure, but flagella are longer and less numerous than cilia. So those are the two basically distinguishing characteristics that you want to remember about the difference between cilia and flagella. Cilia are shorter and more numerous, and flagella are longer, much longer, and less numerous. So some cells, for example, only have one flagellum. Others have multiple, but still not as many as we find cilia on ciliates. So as I mentioned, even though they are different in their length and the number of them, they still have that 9+2 arrangement. They reproduce asexually by mitotic binary fission. So basically, protozoans in general do. Some of them are parasitic. I don't want you to get the idea that all zooflagellates or zoomastiginids cause disease, because they don't. But it's important to us that they are parasites and that they do cause disease, that they are pathogens. So that's why we're looking at pretty much nothing but pathogens on this slide. So we've got the disease, the sexually transmitted disease, the STD, trichomoniasis, commonly known as trich. We've got the waterborne illness, giardiasis. Waterborne meaning that you can get it by drinking water that's contaminated with the giardia organism. Leishmaniasis, which is transferred from person to person by the by the sandfly. So in that way, the sandfly is acting as a vector, what we call a vector. And the same for African sleeping sickness. The tsetse fly transmits it from person to person. The tsetse fly will bite one person that has the disease and transfer the trypanosome organism to the next person that the fly bites. And that person then will contract African sleeping sickness. So the tsetse fly, again, is acting as a vector. So vector, in this sense, refers to something, an agent, for lack of a better word, an agent, an individual, a species that carries an infectious pathogen into another organism, or from one individual to another individual. And then the final thing here is to note that not all zoomastiginids are pathogens. As I said, most are actually free-living in fresh water. But we're looking at diseases here because that's what's important to us. And that's where we'll stop. We'll take a look at the next phylum next time.